Hello everyone, welcome back to CHAP-16 Study Zone. Today we are going to revise a very important and interesting chapter from class 11, excretory products and their elimination. Now don't worry, okay? We are not going to make this chapter boring and stressful. Just keep your book open if you have it with yourself and then relax your mind and revise along with me. If you don't have, then also relax. You just listen to it carefully. Think of this session as a quick study partner who will help you save time and go through the chapter properly. Our body is doing cleaning work every second, and in this chapter, we will see how beautifully it removes waste and keeps us healthy. So get ready, stay focused, and let's revise this chapter together in an easy and fun way. Let's begin. Class 11, Chapter 16, Excretory Products and Their Elimination. Animals accumulate ammonia, urea, uric acid, carbon dioxide, water, and ions like sodium, potassium, chlorine, phosphate, sulfate, etc., either by metabolic activities or by other means like excess ingestions. These substances have to be removed totally or partially. In this chapter, you will learn the mechanism of elimination of these substances with special emphasis on common nitrogenous waste. Ammonia, urea, and uric acids are the major form of nitrogenous waste excreted by the animals. Ammonia is the most toxic form and requires large amount of water for its elimination, whereas uric acid, being the least toxic, can be removed with the minimum loss of water. The process of excreting ammonia is ammonotalism. Many bonifish, aquatic amphibians, and aquatic insects are amniotic in nature. Ammonia, as it is readily soluble, is generally excreted by diffusion across body surface or through gill surface in fish as ammonium ions. Kidneys do not play any significant role in its removal. Terrestrial adaptation necessitated the production of lesser toxic nitrogenous waste like urea and uric acid for conservation of water. Mammals, many terrestrial amphibians, and marine fishes mainly excrete urea and are called ureotelic animals. Ammonia produced by metabolism is converted into urea in the liver of these animals and released into the blood, which is filtered and excreted out by the kidneys. Some amount of urea may be retained in the kidney matrix of some of these animals to maintain a desired osmolality. Reptiles, birds, land snails, and insects excreted nitrogenous waste as uric acid in the form of pellets or paste with a minimum loss of water are called uricotelic animals. A survey of animal kingdom presents a variety of excretory structures. In most of the invertebrate, these structures are simple tubular form, whereas vertebrates have complex tubular organs called kidney. Some of their structures are mentioned here. Protonephridia or phlegm cells are the excretory structure in Platyhelminthes, that is, flatworm, example planaria. Rotifer, some annihilates, and the cephalochordates, Amphioxus. Protonephridia are primarily concerned with ionic and fluid volume regulation, that is, osmoregulation. Nephridia are the tubular excretory structure of earthworm and other annihilates. Nephridia help to remove nitrogenous waste and maintain a fluid and ionic balance. Malpighian tubules are the excretory structure of most of the insects, including cockroaches. Malpighian tubules help in the removal of nitrogenous waste and osmoregulation. Antennal glands or green glands perform the excretory function in crustaceans like prawns. Human excretory system. In human, the excretory system consists of a pair of kidney, one pair of ureter, a urinary bladder, and a urethra. Kidney are reddish brown, bean-shaped structure situated between the level of last thoracic and third lumbar vertebrae, close to the dorsal inner wall of the abdominal cavity. Each kidney of an adult human measure 10 to 12 centimeter in length, 5 to 7 centimeter in width, 2 to 3 centimeter in thickness, with an average weight of 120 to 170 gram. Towards the center of the inner concave surface of the kidney is a notch called hilum, through which ureter, blood vessels, and nerve enter. Inner to the hilum is a broad funnel-shaped space called the renal pelvis, with projection called callus. The outer layer of kidney is a tough capsule. Inside the kidney, there are two zones, an outer cortex and the inner medulla. The medulla is divided into a few conical masses, medullary pyramids, projecting into the calyx. Singularly, we can call it calyx. The cortex extends in between the medullary pyramids as renal column called columns of Bertini. Each kidney has nearly 1 million complex tubular structure called nephrons, which are the functional unit. Each nephron has two parts, the glomerulus and the renal tubules. Glomerulus is a tuft of capillaries formed by the efferent arterioles, a fine branch of renal artery. Blood from the glomerulus is carried away by an efferent arterioles. The renal tubule begin with a double-walled cup-like structure called Bowman's capsule, which encloses the glomerulus. Glomerulus, along with Bowman capsule, is called the Malpighian body or renal corpuscles. The tubule continue further to form a highly coiled network, proximal convoluted tubule. A hairpin shaped Henle's loop is the next part of the tubule, which has a descending and an ascending limb. The ascending limb continues as another highly coiled tubular region called distal convoluted tubule. The distal convoluted tubule DCT of many nephrons open into a straight tube called collecting duct, many of which coverage and open into the renal pelvis through medullary pyramid in the calyx. The Malpighian corpuscles PCT and DCT of the nephron are situated in the cortisol region of the kidney, whereas the loop of Henle dips into the medulla. In majority of nephrons, the loop of Henle is too short and extended only very little into the medulla. Such nephrons are called cortical nephrons. In some of the nephrons, the loop of Henle is very long and run deep into the medulla. These nephrons are called juxtamedullary nephrons. The efferent arterioles emerging from the glomerulars from a fine capillary network around the renal tubule called the peritubular capillaries are minute vessels of this network runs parallelly to the Henle's loops, forming a U-shaped vasa recta. Vasa recta is absent or highly reduced in cortical nephrons. Then urine formation. Urine formation involves three main processes, namely glomerular filtration, reabsorption, and secretion, that takes place in different parts of the nephron. The first step is urine formation, is the filtration of blood, which is carried out by the glomerulus and is called glomerular filtration. On an average, 1100 to 1200 milliliter of blood is filtered by the kidney per minute, which constitute roughly 1/5 of the blood pumped out by each ventricle of the heart in a minute. The glomerular capillary blood pressure causes filtration of blood through three layers, that is, the endothelium of glomerular blood vessels, the epithelium of Bowman's capsule, and a basement membrane between these two layers. The epithelial cells of Bowman's capsule, called podocytes, are arranged in an intricate manner so as to leave some minute space called filtration slits or slit pore. Blood is filtered so finely through this membrane that almost all the constituent of the plasma except the protein passes onto the lumen of the Bowman's capsule. Therefore, it is considered as a process of ultrafiltration. The amount of the filter formed by the kidney per minute is called glomerular filtration rate, GFR. Glomerular filtration rate in a healthy individual is approximately 125 milliliter per minute, that is 180 liter per day. The kidney have built in mechanism for the regulation of glomerular filtration rate. One such efficient mechanism is carried out by juxtaglomerular apparatus, JGA. Juxtaglomerular apparatus is a special sensitive region formed by cellular modification in a distal convoluted tubule, DCT, and the efferent arteriole at the location of their contact. A fall in GFR can activate the JG cells to release renin, which can stimulate the glomerular blood flow and thereby the GFR back to normal. A comparison of the volume of the filtered formed per day, 180 liter per day, with that of the urine release 1.5 liters suggests that nearly 99% of the filtrate has to be reabsorbed by the renal tubule. This process is called reabsorption. The tubular epithelial cell in different segments of nephron perform this either by active or passive mechanism. For example, substances like glucose, amino acid, sodium, etc. in the filtrate are reabsorbed actively, whereas the nitrogenous waste are absorbed by passive transport. Reabsorption of water also occurs passively in the initial segment of the nephron. During urine formation, the tubular cells secrete substances like hydrogen, potassium, and ammonium into the filtrate. Tubular secretion is also an important step in urine formation as it helps in the maintenance of ionic and acid-base balance of body fluids. Function of the tubules. Proximal convoluted tubule PCT is lined by simple cuboidal brush border epithelium, which increases the surface area of reabsorption. Nearly all of the essential nutrient and 70 to 80% of electrolytes and water are reabsorbed by this segment. PCT also helps to maintain the pH and ionic balance of the body fluid by selective secretion of hydrogen ion and ammonium into the filtrate and by reabsorption of bicarbonate from it. Henle's of loop. Reabsorption is minimum in its ascending limb. However, this region play a significant role in the maintenance of high osmolarities of medullary interstitial fluid. The descending limb of loop of Henle is permeable to water but almost impermeable to electrolytes. This concentrate the filtrate as it moves down. The ascending limb is impermeable to water but allows transport of electrolytes actively or passively. Therefore, as the concentration filtrate passes upward, it gets diluted due to the passage of electrolytes to the medullary fluid. Distant convoluted tubule. DCT. Conditional reabsorption of sodium and water takes place in this segment. DCT is also capable of reabsorption of bicarbonate and selective secretion of hydrogen and potassium ion and ammonia to maintain the pH and sodium potassium balance in blood. Collecting duct. This duct extends from the cortex of the kidney to the inner part of the medulla. Large amount of water could be reabsorbed from this region to produce a concentrated urine. This segment allows passage of small amount of urea into the medullary interstitium to keep up the osmolarity. It also plays a role in the maintenance of pH and ionic balance of blood by the selective secretion of hydrogen and potassium ion. Mechanism of concentration of the filtrate. Mammals have the ability to produce a concentrated urine. The Henle's loop of vasa recta play a significant role in this. The flow of filtrate in the two limbs of Henle's loop is in opposite direction and thus form a countercurrent. The flow of blood through the two limbs of vasa recta is also a countercurrent pattern. The proximity between the Henle's loop and vasa recta as well as the countercurrent in them helps in maintaining and increasing osmolarity towards the inner medullary interstitium, that is from 300 milliosmoliter in the cortex to about 1200 milliosmoliter in the inner medulla. This gradient is mainly caused by NaCl and urea. NaCl is transported by the ascending limb of Henle's loop, which is exchanged with the descending limb of vasa recta. NaCl is returned to the interstitium by the ascending portion of vasa recta. Similarly, small amount of urea enter the thin segment of the ascending limb of Henle's loops, which is transported back to the interstitium by the collecting tubules. The above described transport of substances facilitated by the special arrangement of Henle's loop and vasa recta is called the countercurrent mechanism. This mechanism helps to maintain a concentration gradient in the medullary interstitium. Presence of such interstitial gradient helps in an easy passage of water from the collecting tubules, thereby concentrating the filtrate urine. Human kidney can produce urine nearly 4 times concentrated than the initial filtrate form. Regulation of kidney function. Functioning of the kidney is efficiently monitored and regulated by the hormonal feedback mechanism involving the hypothalamus, JGA, and to a certain extent the heart. Osmoreceptors in the body are activated by changes in blood volume, body fluid volume, and ionic concentration, and excessive loss of fluid from the body can activate this receptor, stimulate the hypothalamus to release antidiuretic hormone ADH or vasopressin from the neurohypophysis. ADH facilitates water reabsorption from latter part of the tubule, thereby preventing diuresis, and increasing in body fluid volume can switch off the osmoreceptors and suppress the ADH release to compete the feedback. ADH can also affect the kidney function by its constrictory effect on blood vessels. This causes an increase in blood pressure. An increase in blood pressure can increase the glomerular blood flow and thereby the GFR. The JGA plays a complex regulatory role. A fall in glomerular blood flow, glomerular blood pressure, GFR can activate it and JG cells to release renin, which convert angiotensinogen in blood to angiotensin 1 and further to angiotensin 2. Angiotensin 2 begin a powerful vasoconstrictory, increase the glomerular blood pressure and thereby GFR. Angiotensin 2 also activate the adrenal cortex to release aldosterone. Aldosterone causes reabsorption of sodium and water from the distal part of the tubule. This also leads to an increase in blood pressure and GFR. This complex mechanism is generally known as the renin-angiotensin mechanism. An increase in blood flow to the atria of the heart can cause the release of atrial natriuretic factor ANF. ANF can cause vasodilation, dilation of blood vessel, and thereby decrease the blood pressure. ANF mechanism therefore act as a check on the renin-angiotensin mechanism. Urine formed by the nephron is ultimately carried to the urinary bladder, where it is stored till a voluntary signal is given by the central nervous system CNS. This signal is initiated by the stretching of the urinary bladder. As it gets filled with urine, in response, the stretch receptor on the walls of the bladder sends signals to the CNS. The CNS passes on motor messages to initiate the contraction of smooth muscles of the bladder and simultaneous relaxation of the urethral sphincter, causing the release of urine. The process of release of urine is called micturations, and the neural mechanism causing it is called the micturation reflex. An adult human excrete on an average 1 to 1.5 liter of urine per day. The urine formed is a light yellow colored watery fluid which is slightly acidic, pH 6.0, and has a characteristic odor. On an average, 25 to 30 gram of urea is excreted out per day. Various conditions can affect the characteristics of urine. Analysis of urine helps in clinical diagnosis of many metabolic disorders, well as malfunctioning of the kidney. For example, presence of glucose, glycosuria, and ketone bodies, ketonuria, in urine are indicative of diabetes mellitus. Role of other organs in excretion. Other than the kidney, lungs, liver, and skin also help in the elimination of excretory waste. Our lungs remove large amount of carbon dioxide, approximately 200 milliliter per minute, and also significant quantities of water every day. Liver, the largest gland of in our body, secrete bile-containing substances like bilirubins, biliverdin, cholesterol, degraded steroid hormones, vitamins, and drugs. Most of these substances ultimately pass out along with digestive waste. The sweat and sebaceous glands in the skin can eliminate certain substances through their secretion. Sweat produced by the sweat gland is a watery fluid contain sodium chloride, NaCl, small amount of urea, lactic acid, etc. Through the primary function of sweat is to facilitate a cooling effect on the body surface. It also helps in the removal of some of the waste mentioned above. Sebaceous gland eliminates certain substances like sterols, hydrocarbons, and waxes through sebum. This secretion provides a protective oily covering from the skin. Do you know that small amount of nitrogenous waste can be eliminated through saliva too? Disorder of the excretory system. Malfunctioning of kidney can lead to the accumulation of urea in blood, a condition called uremia, which is highly harmful and may lead to kidney failure. In such patients, urea can be removed by a process called hemodialysis. During the process of hemodialysis, a blood drained from a convenient artery is pumped into a dialysis unit called artificial kidney. Blood drained from a convenient artery is pumped into a dialysing unit after adding an anticoagulant like heparin. The unit contains a coiled cellophane tube surrounded by a fluid, dialytic fluid, having the same composition as that of plasma except the nitrogenous waste. The porous cellophane membrane of the tube allows the passage of molecule based on concentration gradient. As nitrogenous waste are absent in the dialysing fluid, these substances freely move out, thereby clearing the blood. The clear blood is pumped back to the body through a vein after adding antiheparin to it. This method is borne for thousands of uremic patients all over the world. Kidney transplantation is the ultimate method in the correction of acute renal failure, kidney failure. A functioning kidney is used in transplantation from a donor, preferably a close relative, to minimize its chances of rejection by the immune system of the host. Modern clinical procedures have increased the success rate of such a complicated techniques. Renal calculi, stone or insoluble mass of crystallized salt, oxalates, etc., formed within the kidney. Glomerulonephritis, inflammation of glomeruli of kidney. Summary. Many nitrogenous containing substances and carbon dioxide, water, etc. that accumulate in the body have to be eliminated. Nature of nitrogenous waste formed and their excretion vary among animals, mainly depending on the habitat, availability of water. Ammonia, urea, and uric acid are the major nitrogenous waste excreted. Protonephridia, nephridia, Malpighian tubules, green glands, and the kidney are the common excretory organs in animals. They not only eliminate nitrogenous waste, but also help in the maintenance of ionic and acidic base balance of body fluid. In human, the excretory system consists of one pair of kidney, a pair of ureter or urinary bladder, and a urethra. Each kidney has over a million tubular structures called nephrons. Nephron is the functional unit of kidney and has two portions, glomerulus and renal tubules. Glomerulus is a tuft of capillary formed from efferent arterioles, fine branches of renal arteries. The renal tubules start with a double-walled Bowman's capsule and is further differentiated into proximal convoluted tubule PCT, Henle's loop HL, and distal convoluted tubule DCT. The distal convoluted tubules of many nephrons join to a common collecting duct, many of which ultimately open into the renal pelvis through the medullary pyramid. The Bowman's capsule encloses the glomerulus to form Malpighian or renal corpuscles. Urine formation involves three main processes, that is filtration, reabsorption, and secretion. Filtration is a non-selective process performed by the glomerulus using the glomerular capillary blood pressure. About 1200 milliliter of blood is filtered by the glomerulus per minute to form 125 milliliter of filtrate in the Bowman's capsule per minute. JGR, a specialized portion of the nephron, play a significant role in the regulation of JFR. Nearly 99% reabsorption of the filtrate takes place through different part of the nephron. PCT is the measure site of reabsorption and selective secretion. HL primarily, Henle's of loop primarily helps to maintain osmolar gradient 300 milliosmoliter to 1200 milliosmoliter within the kidney interstitium. DCT, distal convoluted tubule, and the collecting duct allow extensive reabsorption of water and certain electrolytes which help in osmoregulation. Hydrogen, potassium, and ammonia. could be secreted into the filtrate by the tubule to maintain the ionic balance and pH of body fluid. A countercurrent mechanism operates between the two limbs of the loops of Henle's and those of vasa dicta. Capillary parallel to Henle's loop, the filtrate gets concentrated as it moves down the descending limb but is diluted by the ascending limb. Electrolytes and urea are retained in the interstitium. By this arrangement, density and collecting duct concentrate the filtrate about 4 times, that is from 300 milliosmoliter to 1200 milliosmoliter. An excellent mechanism of conservation of water, urine is stored in the urinary bladder till a voluntary signal from CNS carries out its release through urethra, that is micturition. Skin, lungs and liver also assist in excretion. And that brings us to the end of this chapter. I hope this book reading session helped you revise the chapter in an easy and clear and time-saving way. So now give yourself a little smile because one more important chapter is done. Kudos to you. Do not forget to quickly go through the important points once again, okay? Especially the key terms and diagrams and the processes like urine formation and the filtration term, okay? So if this session helped you, please like the video, share it with your friends and subscribe to the channel for more simple and stress-free revision session. Keep learning, keep revising and see you in the next chapter. Bye-bye. Take care.